Hello and welcome to Dear Doctor, the channel where we explore the latest and most fascinating medical topics. Today we're going to talk about a new discovery that could revolutionize the treatment of spinal cord injury. Spinal cord injury, or SCI, is a devastating condition that affects millions of people worldwide. It occurs when the spinal cord, the bundle of nerves that connects the brain to the rest of the body, is damaged by trauma, disease, or infection. This damage can interrupt the communication between the brain and the body, resulting in loss of sensation, movement, or function below the level of injury. Currently, there is no cure for SCI, and most treatments focus on preventing further damage, managing complications, and improving quality of life. However, there is hope for a better future, thanks to a breakthrough discovery made by a team of researchers from Harvard University and Boston Children's Hospital. The researchers have uncovered a crucial component for restoring functional activity after SCI. They have successfully regenerated specific neurons in mice, leading to restored walking ability. Their findings were published in the journal Nature on October 13, 2023. The researchers focused on a type of neuron called corticospinal motor neurons, or CSMNs. These neurons are responsible for controlling voluntary movements, such as walking, running, or grasping. They originate in the cerebral cortex, the outer layer of the brain, and extend their axons, or long projections, down the spinal cord to connect with other neurons or muscles. After SCI, CSMNs are among the most vulnerable neurons to die or degenerate. This is because they have very low levels of intrinsic growth potential, meaning they are unable to regenerate their axons and reconnect with their targets. This leads to permanent loss of motor function below the level of injury. However, the researchers discovered that CSMNs can be reprogrammed to enhance their growth potential and regenerate their axons after SCI. They did this by manipulating a gene called pten, which normally acts as a brake on cell growth and survival. By deleting pten specifically in CSMNs using genetic engineering techniques, they were able to boost their growth potential and survival rate. The researchers then tested whether these reprogrammed CSMNs could regenerate their axons and restore function after SCI in mice. They induced SCI in mice by cutting their spinal cords at the thoracic level, which paralyzes their hind limbs. They then injected a virus that carries the ptendeletion gene into the cerebral cortex of some mice, while leaving others as controls. After 4 weeks, they observed that the mice with ptendeletion had significantly more CSMNs surviving and regenerating their axons across the injury site than the control mice. Moreover, these regenerated axons were able to form functional connections with other neurons or muscles below the injury site. The researchers then assessed whether these regenerated connections could translate into improved motor function in the mice. They used various behavioral tests to measure the locomotor ability of the mice, such as walking on a treadmill or crossing a horizontal ladder. They found that the mice with ptendeletion had significantly better walking performance and coordination than the control mice. These results demonstrate that ptendeletion can enhance CSMN regeneration and functional recovery after SCI in mice. This is a remarkable achievement that opens new possibilities for treating SCI in humans. Of course, there are still many challenges and questions that need to be addressed before this approach can be translated into clinical trials. For example, how safe and effective is ptendeletion in humans? How can ptendeletion be delivered precisely and efficiently to CSMNs in humans? How can ptendeletion be combined with other therapies, such as stem cells or electrical stimulation? How can ptendeletion be applied to different types and severities of SCI? These are some of the questions that the researchers are working on to further advance this promising field of research. That's all for today's episode of Dear Doctor. I hope you enjoyed it and learned something new. If you did, please give this video a thumbs up and subscribe to our channel for more medical topics. And don't forget to leave your comments and questions below. I'll see you next time on Dear Doctor.